everybody. My name is Alex Muir from the University of Chicago. I want to start by thanking the organizers and lab roots for putting together this 13th cancer research series. And I'm super excited to be here to tell you about work from our group looking at the role of metabolic stress in tumor biology. Or how we like to think about this in our lab is cancer cells frequently are in a hangry cell state. And we think that knowledge of this hangry cell state is really going to be critical for better understanding and targeting cancers. So I'll start by talking about why tumors are metabolically stressed out. So in order to discuss this, we first need to know how healthy tissues are fed. So healthy tissues are fed from nutrients that come into a tissue via the circulatory system. They enter a capillary bed, and the capillary bed in the tissue acts as a filtration system to keep blood cells and protein in the circulatory system, but to filter out a nutrient-rich fluid called interstitial fluid into the microenvironment of the tissue. And this interstitial fluid is actually what brings nutrients and carries waste away from cells in a tissue. Now, this capillary system and filtration system is normally very efficient, such that the metabolic microenvironment of a tissue is roughly in equilibrium with what's going on in the bulk circulation. So if we look at skin as an example, if you look at skin interstitial fluid, the concentration of glucose in the microenvironment of skin versus venous glucose, so the glucose concentration in the bulk circulation. In humans, there's normally a one-to-one correlation between these measurements, really indicating that the metabolic microenvironment of tissues is roughly in equilibrium with what's going on in the bulk circulation. Now, for tumors, we have reason to believe that this is not likely to be the case. A hallmark of many solid cancers is aberrant angiogenic signaling, and this results in vasculature in tumors, which is really quite compromised compared to healthy tissues. Many tumors are hypovascularized, and the blood vessels that are there are disorganized and dysfunctional. And this can lead to poor perfusion, poor delivery of nutrients to tumors compared to healthy tissues. And we can experimentally see this using perfusion tracers. An example here is the dye Huxt, which can be delivered to the circulatory system of tumor-bearing or healthy animals. And Huxt, as it transits through the body, if a cell in a tissue is well perfused, it'll take up Huxt and its nuclei will become labeled. In contrast, a cell that is not well perfused won't take up this dye and will be Huxt negative. Now, if we perform this perfusion tracing in a healthy animal with the normal pancreas, you can see that every cell in the pancreas is taking up Huxt in the cyan color, indicating that the whole pancreas is well perfused. In contrast, if we do the same labeling experiment from a pancreatic ductal adenocarcinoma or PDAC tumor, this is the type of tumor that we work with in our group, so you'll see a lot of data from these PDAC tumors throughout the talk. You can see that many of the cells in these PDAC tumors are Huxt negative, indicating that the vast majority of cells in these cancers are poorly perfused. And so the fact that tumors are hypovascularized, their vasculature is dysfunctional, and we can see that they're poorly perfused. This has led to the hypothesis that tumors likely experience nutrient or metabolic stress. And so what I'm going to talk about for the rest of our time today is some techniques that we developed to go in and look molecularly what the stress actually looks like in a tumor. And then what I want to talk about for the second part of the talk is how we can leverage this information to understand the constraints that cancer cells face and synthetic lethal interactions, new ways to target cancers that arise from these TME or tumor microenvironmental nutrient stresses. All right, so what does this stress look like in a tumor? So I just argued that the microenvironment of a tumor is likely to be out of equilibrium with what's going on in the bulk circulation. So if we want to know the metabolic stresses that a cancer might face, it's not going to be enough to look in the bulk circulation from a plasma draw, for example. We have to go in and look locally at the interstitial fluid from the tumor microenvironment to see the kind of metabolic stresses that are occurring. So a number of years ago now, we adapted some techniques from tissue physiology to isolate tumor interstitial fluid, or TIF, from PDAC tumors. And we're really excited to have these TIFF samples because we think these are a really unique lens on the tumor microenvironment. It's kind of the soluble tumor microenvironment. And by analyzing the composition of what's in the TIFF, we can really enhance our understanding of what the prevailing conditions in the tumor microenvironment are. Of course, we were really interested in what the metabolite levels are in the tumor microenvironment. So once we had these interstitial fluid samples, we developed a mass spec pipeline where we can use really small volumes, but can still perform absolute quantification on more than 180 major circulating nutrients and vitamins, each one of which on this metabolic map is highlighted with a black dot. So we have the interstitial fluid samples now. We have the analytical pipeline where we can measure nutrient concentration. What did we see in the microenvironment of these pancreas cancers? The first thing that I want to highlight is if we compare by principal component analysis, all of the metabolites that we measured in the interstitial fluid versus the plasma, that these readily cluster separately from each other. And so in contrast to healthy tissues where the nutrient concentrations are quite similar to in the tissue microenvironment compared to the bulk circulation, we really see in these pancreas cancers that these are quite distinct. And we think this is very much consistent with this model of healthy tissues, well-organized vasculature, their metabolic microenvironment in equilibrium with the bulk circulation. Cancers, because of their abnormal vasculature, really are build their own unique metabolic microenvironment. All right, so what are the particular nutrient stresses that are prevalent in pancreas cancers? I'll go through some of the data here in a volcano plot comparing concentrations of nutrients and TIFF from a mouse model of pancreas cancer versus plasma, because there's a lot of surprises that updated my mental model for metabolic stress and cancer. The first thing that I really want to highlight here is over here on the right, there's a lot more metabolites that are enriched in TIFF compared to those that are depleted. Some of these make a lot of sense to us. Glutamate and glycine, these are two nutrients that cancer cells secrete in culture to high degrees. Potentially this explains why they accumulate in the microenvironment. But there's a number of other number of other nutrients such as neurotransmitters like GABA, redox molecules like glutathione that are also accumulating in the tumor microenvironment. We're really interested in understanding how some of these things are accumulating and what functional roles they might have. But big picture, what I really want to highlight here is my mental model before we started this work was that the nutrient microenvironment is going to be a desert. There's not a lot of nutrient delivery. You have a lot of hungry cancer cells, so most things are going to be depleted. In fact, there's more things that are enriched in the tumor microenvironment. So I think it's important to think about metabolic stress as not just being a desert, but it's also a jungle of things that are accumulating in tumors as well. Going here to the center of this volcano plot, there's a lot of additional surprises for us as well. There's a lot of surprises here for us as well. So going back to the cell culture studies, there's two nutrients, the sugar, glucose, and the amino acid glutamine, which are the most heavily consumed by cancer cells in culture. So we really thought that these would be the two nutrients that are by far the most depleted in the tumor microenvironment. We in fact didn't find evidence of glutamine depletion, and glucose, while it is depleted, is only depleted to a relatively minor degree. And the byproduct of glucose degradation via glycolysis, lactate, we didn't see evidence for lactate accumulating in the tumor microenvironment as well. And so again, this kind of updated our mental model that perhaps glucose and glutamine, these might not be the two most prevalent metabolic stresses, at least in this pancreas cancer model. So what are the nutrient and metabolic stresses that we observed in these pancreas cancers? The three most prevalent stresses that we saw are depletion of a trio of amino acids, the amino acid arginine, which we'll come back and talk about later in the talk, amino acids cysteine and tryptophan, all three heavily depleted from these pancreas cancers. And so amino acid stress really seems to be a key metabolic stress in these pancreas cancers. And additionally, we also saw strong depletion of this organic acid pyruvate. This is a nutrient that hypoxic cells will consume to help them maintain homeostasis and viability. And so perhaps it makes sense that this would be depleted from these pancreas cancers that are poorly perfused and hypoxic. So I'll stop there in terms of discussing the metabolites that are present in these pancreas cancers. But I just want to highlight that there's been many studies now using this approach for understanding metabolic stresses in diverse tumor types. And we recently put together a review collating kind of all the interstitial fluid metabolite concentrations for all the different tumor types where these studies have been performed. And so if you're interested in, for your particular cancer type that you're interested, understanding the metabolic stresses that are prevalent in that tumor type, strongly urge you to check out this review. Hopefully some helpful data in there for you. All right, so I want to switch gears now, since we now have good data on what nutrient stress actually looks like in these pancreas cancers. How can we leverage this information to better understand how these tumors function and how we can target them? So first, I want to introduce this concept of microenvironmental or TME synthetic lethality. So synthetic lethality is this idea that if you have some essential process, like a central metabolic pathway, if there's two kind of semi-redundant pathways that feed into that process, each one of these, it's tolerable for a cell to lose this. For example, you can lose the red pathway up here, as there's this backup blue pathway that can compensate. Now, if a tumor loses one of these pathways, for example, via genetic lesion, this can render the other pathway a great anti-cancer target, because cancer cells, this will now be synthetically lethal, while healthy cells will be less affected by targeting of this pathway. And for genetic lesions, genetic impairments of different processes in cancer cells, identification and targeting of synthetic lethal pathways, this has been something that we've been leveraging now for decades. But there's also another way in which we can identify these synthetic lethal opportunities. And that's not just by genetic loss of a pathway in a cancer cell. For example, microenvironmental limitations can also constrain a pathway and open up synthetic lethal opportunities as well. And so we have been really interested in trying to identify what are the synthetic lethal targets, what are the constraints on different pathways by nutrient stress in the microenvironment, and what are the synthetic lethal opportunities that arise as a result of these constraints. So this is what I want to talk about now. And the last bit of the talk is, what are the synthetic lethal interactions that arise from the microenvironmental stress that we've seen from our interstitial fluid analyses? So in order to identify these microenvironmental stresses, we first need a way to study cells as they are constrained by and see the nutrient stress of the microenvironment. And so to do this, we took the interstitial fluid concentrations of nutrients that we had, and we built a cell culture media that we call tumor interstitial fluid medium, or TIFM. And this contains 115 metabolites and vitamins at the concentrations that we experimentally observed in the microenvironment of these tumors. And what this media allows us to do is we can go in now and we can isolate different cell types from tumors that we're interested in, and we can culture them in standard conditions or in our TIFM culture. And then we can look at the differences in behavior and biology between these two conditions in order to determine how microenvironmental metabolic stress is impacting and talking to different cell types in tumors. And so to understand how the nutrient stress of the tumor microenvironment was constraining and limiting different pathways in pancreas cancers, we took mouse transcriptional approach. We took mouse pancreas cancer cells and we grew them in 3 different ways. We either grew them in standard culture conditions, and we kind of think about this as the cancer cells on their own, no microenvironment arm of this experiment, because the cells are growing as a monolayer by themselves with no cues from the microenvironment at all. We grew the same cells in our tumor interstitial fluid medium, so the cells are now just seeing nutrient stress of the microenvironment isolated on its own. Or we put these cells orthotopically into the pancreas of a mouse, where they formed a tumor, and so now these cells are experiencing every feature of the tumor microenvironment. We collected cells from all of these conditions and performed transcriptomic analysis and looked at both the upregulated and downregulated pathways. And what we found is in our TIFM arm of the experiment, there's a number of metabolic pathways that were strongly downregulated by nutrient stress. And we were quite interested in these because these might be pathways that are constrained by nutrient stress in the microenvironment that could potentially open up synthetic lethal opportunities. One recurrent set of hits that we got from this transcriptional analysis was SRB, or targets of the SRBP transcription factor. And I'll talk about what SRBP is in a second, but I want to point out before we get to that SRBP is not only downregulated in our TIFM culture model, but it's also downregulated in vivo as well. So if we look at SRBP target genes, these are strikingly downregulated in murine and orthotopic murine PDAC tumors compared to standard culture. And we think this is occurring in human pancreas cancer as well, not just mouse biology. So in collaboration with Meadows Gurzis and John Koloff at UIC, we compared the transcriptional profile of primary pancreas cancers compared to, so these are pancreas cancer cells that are seeing every aspect of the microenvironment, including nutrient stress. Or we looked at pancreas cancer cell lines. So again, these are cancer cell lines kind of on their own, growing in a dish. And when we look at expression of SRBP target genes, in primary tumors, these are really strongly downregulated compared to just the cancer cells isolated on their own. So both in human and in murine PDAC specimens, we think that the tumor microenvironment, nutrient stress in particular, is strongly suppressing SRABP. All right, so what is SRABP? SRABP is a transcription factor that is a strong activator of lipogenesis, which is a way that cells can, one of two ways that cells have to get the lipids that they need for homeostasis. Cells can either make their own lipids via lipogenesis, or they can take up lipids from the environment. SRBP is really critical for this lipogenesis pathway. And so our model is that metabolic stress in the tumor microenvironment is suppressing SRBP on the basis of this transcriptional data. So first we wanted to ask what particular metabolic stresses are responsible for inhibiting SRBP. So our tumor interstitial fluid medium is chemically defined, and this allows us to drill down and identify the particular metabolic stresses that drive a phenotype of interest, in this case, suppression of SRBP. What we found is that arginine deprivation in particular is responsible for SRBP suppression. So I mentioned, and we were talking about the TIFF measurements, that arginine is one of the most strikingly depleted amino acids in the tumor microenvironment. In plasma, it's present at a concentration of around 120 micromolar. In the tumor microenvironment, this drops about 50-fold, so it's only present at 2 to 5 micromolar. And if we look at SRBP protein expression by immunoblotting, in standard arginine-rich conditions, you can readily detect this protein. It's strongly downregulated in our arginine-deprived TIFM conditions. If we lower arginine concentrations in standard media to what we see in the microenvironment, SRBP is strongly downregulated. In contrast, if we take our tumor interstitial fluid media and supplement additional arginine, this largely restores SRBP expression. And so we know that arginine starvation in these tumors is responsible for suppressing SRBP. All right, so we now know that we have arginine depletion, and this is constraining a particular pathway in pancreas cancer cells. It suppresses SRBP and lipid synthesis. And so we were really interested in understanding what are potential synthetic lethal targets that arise on the basis of this constraint on lipid synthesis. So one hypothesis that we had is goes back to understanding what a really critical function of lipid synthesis in cancer cells is. So all cells have lipids and membranes that are composed of 3 different kinds of fatty acids: saturated fatty acids that contain no double bonds, monounsaturated fatty acids that contain a single double bond, and polyunsaturated fatty acids that contain 2 or more double bonds. De novo lipid synthesis importantly provides a source, endogenous source of mono and saturated fatty acids. And this source of saturated fats and monounsaturated fats is really critical, could be really critical for allowing cells to buffer high levels of a particular fatty acid in the microenvironment. For example, if you have a large bolus of PUFAs in the microenvironment that a cell might take up via uptake, this is okay because the cells can synthesize their own saturated and monounsaturated fats to balance out the fatty acid content in the membrane. We were really interested and hypothesized potentially because arginine restriction suppresses de novo lipid synthesis, this might make cells really susceptible to exogenous polyunsaturated fats because they might end up with not having a source of mono and saturated fats to balance out all these polyunsaturated fats, and could end up with toxic PUFA enrichment in their membranes. And so to test this, we took cells that were cultured in arginine in our TIFM, arginine restricted conditions, or an RPMI, and we cultured them with a series of different polyunsaturated fatty acids. And in every case, what we found is cells in arginine-rich conditions are largely unaffected. They're able to grow, it's measured by doublings per day here, and the presence of these polyunsaturated fatty acids. In contrast, the cells that are in our arginine starvation conditions are extremely sensitive to these same polyunsaturated fats. And we think that this is due to inability to buffer these polyunsaturated fatty acids in the membrane, in the lipidome, because in collaboration with Evan Lien and Kelly Sokol at the Van Andel Institute, we performed lipidomic analysis on cells that were cultured either in TIFM with or without supplemental arginine. And these cells were treated with this polyunsaturated fat, alpha L-eosteric acid. And in arginine starvation conditions, if we give these cells supplemental polyunsaturated fat, we see a high degree of enrichment of this polyunsaturated fatty acid in phospholipids and triglycerides. And just by adding supplemental arginine, the amount of PUFA that ends up in these phospholipids and triglycerides is really strongly suppressed. Very much consistent with this arginine starvation, preventing cells from buffering their membranes from high degrees of polyunsaturated fats. We also wanted to ask if this is true of pancreatic tumors as well. And so to ask this, we first need a way to manipulate levels of arginine in the microenvironment. So why is arginine really strongly depleted from these pancreas cancers? Well, pancreas tumors recruit a large number of arginase-expressing macrophages. You can see here from IHC staining for ARG1. In murine and human PDAC, there's a large number of arginase-positive cells. And arginase is an enzyme that degrades arginine to ornithine and urea. And so by knocking out arginase from myeloid cells, we can manipulate levels of arginine in the tumor. So we made a mouse model where we have deletion of arginase under the control of a myeloid-specific CRE. We can implant pancreas tumors into these mice, and in order to manipulate the arginine levels. So control animals will have plenty of arginase expressing myeloid cells. They'll have an arginine depleted tumor microenvironment. And our myeloid ARG1 knockout mice have arginase deleted from this compartment, and arginine levels are now restored in the tumor microenvironment. And so we took these animals where we've manipulated arginine levels, and we fed them either a control diet or a PUFA-enriched diet. And what we found is in control mice, where arginine is depleted in the tumor microenvironment, the PUFA oil, like we saw in vitro, suppresses the growth of these pancreas cancers. And we were really excited to see a report from Christian Raziz in Mandar Mazumdar's lab at Yale, which also found that pancreas cancers are susceptible to PUFA-enriched diets. So very consistent with our findings. When we did the same feeding experiment now in our myeloid ARG knockout mice, where we've restored arginine levels, the impact of this diet is now lost, consistent with SRBP being turned back on and tumors being able to buffer this bolus of polyunsaturated fat. And again, in collaboration with Kelly and Evan at the Van Andel Institute, we performed lipidomic analysis. And in control tumors, when we fed the PUFA-rich oil, we see an increase in the cellular peroxidation index. This is a metric for how many PUFAs basically are in the lipidome. You see a strong increase in the amount of in the CPI of these specimens. In contrast, if we do the same feeding experiment in our myeloid ARG knockout animals, this increase is now lost, consistent with the ability of these cells to be able to buffer these polyunsaturated fats. All right, so I'll stop there and some of our key findings. The first thing that I really want to highlight is that metabolic stress is a key feature of the microenvironment in many, many solid tumors. There's poor perfusion and resulting metabolic stress. This is really something that is quite a common feature. And this metabolic stress, we're very excited that our data is starting to suggest this can lead to targetable vulnerabilities that we can leverage via this kind of microenvironmental synthetic lethality approach. So as we identify the TME limitations and the pathways that these limitations suppress, we can now go in and identify new synthetic lethal targets that we just wouldn't have appreciated before having this information on what the metabolic stresses in the tumor microenvironment actually are. And the example I want to highlight from our work today is we found that arginine depletion really suppresses lipid synthesis in these pancreas cancers. And the compensatory pathway that these cells have is lipid uptake. And we can target this by feeding cells polyunsaturated fats to overwhelm them basically with all of these PUFAs and lead them to loss of membrane homeostasis and cell death. More broadly, I really want to highlight that this kind of interstitial fluid analysis can really provide a lot of insight into what the different stresses and constraints of the tumor microenvironment are. I've shown some of the data that we've looked at, seeing the metabolic stresses that occur in these tumors. But I think as we use different analytical platforms, get a bigger, broader picture of what the interstitial fluid composition looks like, we'll find a lot out about what the microenvironment of tumors looks like. And lastly, an area of growth that I think I really want to highlight and really challenge folks to think about creative ways that we can overcome this barrier is we can make a lot of measurements of what is present in interstitial fluid now, but it's going to be really critical to be able to match up those measurements of things that we see in the tumor microenvironment to effects on different cell types in the tumor. And so coming up with creative ways to start to match cell biology to features of the microenvironment, I think is going to be really critical. One approach that we took is building a model system that incorporates basically stuff that we saw in the tumor microenvironment so we can study how that impacts cancer cells. That's one approach, but I think we're going to need a lot of creative thinking in this area to fully leverage all of the information that we're getting on the tumor microenvironment. And with that, I'll stop and want to say a big thank you to everybody in our group. In particular, I want to highlight Patrick Yonker, who did a lot of the lipid work from our team. I want to say a big thank you to our collaborators, Meta and John at UIC, who helped us with the patient analysis of PDAC tumors, Evan and Kelly at Van Andel, who helped us with all the lipidomic experiments that we did as part of this study, and our collaborators at UChicago, Hardik, Kay, and Chris. I'll say a big thank you to our funding sources, and I will stop there. Thank you very much.